1-D/D/98. Biology has always received accreditation A from National Accreditation Board for Higher Education and has also been certified ISO 901-2015 in 2018. In the efforts to promote biology study programs to the international level, the Biology implement a quality assurance system that meets the national and international standard, conduct bilingual classes and a number of courses, send students to conduct practicum in several Asian countries, conduct international research collaborations, and publish scientific articles in international journals. Recently, the management of teaching staff at both biology education and biology study program is managed by Department of Biology Education. The total number of lectures is 34. The data of lecture by academic positions and level of education are presented in the diagram. Lecturers at biology study programs are members of certain expertise groups and actively carry out research and community service and publish the result in the form of books, national and international articles, as well as patent certificates and intellectual property rights. Biology study programs offers a flexible curriculum, allowing students to choose a range of courses and programs that suit their needs and interests. The curriculum supports the government's policy of independent learning. In developing the curriculum, biology educations take the steps as seen at the pictures. The curriculum of Bachelor Program of Biology pays attention to the balance between attitudes, knowledge, and skills. The objective of the BPB is to educate students to become researchers in one of the 3 research groups within BPB and to have entrepreneurial skills that are relevant to biology. To improve the quality and teaching in biology study program, the results of the research are applied in the courses. Research-based teaching materials combine theory and research findings so that the students can understand the implications of the concepts. The research involves students under the research group to propose competitive research grant organized by government or other institutions. Currently, biology study program has around 171 students. To facilitate the development of their competences, interests, and talents, students can take part in a number of students' activities such as Bakti Formika Untuk Bangsa or BFUB, Open Labs, Community Service, Students Mountaineering Club, and national seminars. Various achievements of the students of biology educations, among others, getting a silver medal in the national competition for mathematics and natural sciences (KN NIPA), grants for the students' creativity program (PKM), and the entrepreneurial students program (PMW), student exchange and teaching practices at Asian level. Biology study program collaborates with the industry to develop the research in the field of biology area. In a program of accelerating the completions of studies, lectures involve students in their research projects so that students are helped in terms of research funding and the implementation of more targeted student research because it is following the roadmap of the supervisor. To facilitate the learning and research process, lecturers and students can also use various facilities provided at the Department of Biology Education, namely Physiology Laboratory, Ecology Laboratory, Plant Structure Laboratory, Animal Structure Laboratory, Genetics and Microbiology Laboratory, Research Laboratory for Biotechnology and Environment, and also Botanical Garden Laboratory. After graduating from the Biology Study Program, graduates will obtain an academic degree of Bachelor of Science or SSA. The alumni are scattered in many regions in Indonesia and abroad. Based on the result of the transfer study, practitioners, research assistants, writers, government employees, private employees, educators, and successful entrepreneurs. Currently, Bachelor Program of Biology does not only produce professional research candidates, but it is much more than that, namely to produce superior generations in a complete manner, both in mind and behavior, so that they can provide benefits to society, the nation, and the sustainability of life on Earth.
Okay, thank you very much. Good morning, everyone.
Good morning.
Okay, we're going to start our session today. It's already recorded, right? Okay, good morning. A warm welcome to all participants joining us again in the Adjunct Professor Series organized by the Biology Study Program, Universitas Pendidikan Indonesia. We are very pleased to continue our second session today entitled Microalgae Biotechnology, Deciphering Artificial Intelligence Driven and Prediction Mode.
Iya, 4.000 lah udah kita. Padahal telat, kakak berarti 5.000. Iya, bisa jadi 6.000.
Okay. Yeah, following yesterday's insightful discussion on microalgae biorefinery, today's session will dive deeper into how artificial intelligence and predictive modeling can be applied to optimize microalgae biotechnology, a topic that, as promised by Dr. Khoo yesterday, will be even more exciting and engaging. We are once again honored to welcome Dr. Kuan Shiong, Assistant Professor from Yonsei University, Taiwan, a leading researcher with extensive expertise in microalgae bioprocessing and bioseparation technology. Before we begin, let me kindly inform you that Professor Topik Hidayat, who was originally scheduled to share his material today, will instead deliver his presentation in our third meeting on November 14 with the topic Microalgae-based Future Trees. We truly appreciate your understanding and look forward to that session next week. Okay, before we begin, let me kindly remind everyone of a few guidelines. The first one is please use the virtual background that has been sent to everyone and keep your camera on. And please mute your microphone unless you are speaking. Later, during the discussion session, you are welcome to ask question directly via open mic or type the question in the chat box. Okay, now to begin today's program, we will first have a group photo session together again. So please open your camera, everyone, and prepare your smile. Okay. Please open your camera. Okay. Okay, since we don't know our location in the Zoom, just hold your smile again until we finish this session. Okay, Ibu Nur Fitri will help to capture the moment. Ibu Fitri siap?
Okay,
Okay. The
so
first page.
the first slide,
One.
2,
Okay.
3.
Okay.
And second slide. And the last one.
Okay. All captured, Bu Nur Fitri?
Yes, all culture.
Okay, thank you very much. And now we continue to the main session. But before that, because the participant is different with the previous one, so I'll going to read again or introducing again a brief of Dr. Khoo's CV. But Afri, can you please share the screen? Okay, can you please zoom in to the CV? Okay. So our speaker today, Dr. Khoo, is an assistant professor at the Department of Chemical Engineering and Material Science, UNC University, Taiwan. He is an expert in microalgae biorefinery, focusing on both upstream and downstream processing, as well as the integration of artificial intelligence and Internet of Things in bioprocess engineering. Dr. Khoo earned his bachelor degree in applied chemistry from University Technology Petronas, Malaysia, and completed his PhD in chemical and environmental engineering at the University of Nottingham, Malaysia, within just 2 years. He has published over 200 scientific papers with more than 10,000 citations and an H index of 65, and is recognized among the top 20% world scientists by Stanford University in 2023 and 2024. He is also active as adjunct and visiting professor in several universities across Asia. Okay, thank you, Pak Afri. Okay, amazing, guys. So, okay, without any further ado, let's jump into our main session of presentation from Dr. Kuo. Let's enjoy this session and gain new insight into the of microalgae biotechnology and artificial intelligence. Please welcome Dr. Kuan Shiong Kuo.
Okay, thank you so much, Miss Nadia, for the very warm introduction. Um, so everyone can see my screen. Can you all see my share screen?
Yes, we can see it.
Okay, that's great. Okay, so good morning, everyone. I think I heard that Miss Nadia mentioned that the current the current students here today are participants here today are different, slightly different compared to the one in the first class, okay, the first workshop. But anyways, I'm very grateful to be here today, and I hope that for today's topic, which is on artificial intelligence, and I'm going to share how am I going to use artificial intelligence AI as a prediction model and how to use it into microalgae biotechnology. Okay, so my name is Guan Xiong. I hope you guys will enjoy the presentation. Okay, so first thing first, I would like to introduce a very brief overview. What is actually AI-driven technology? Okay, basically AI, artificial intelligence, they are sets of what we call quantification methods, okay, that we can use to quantify the growth of the microalgae. For example, you can see that from this image, right? The first thing is that we cultivate the microalgae, we do some sampling, all right? But the conventional way of doing it is that we will use the UV spectrophotometer. But one thing is that if we are using a lot of equipment to analyze the microalgae, one thing we notice is that it may not be very sustainable, or maybe the laboratorious work is quite a lot. And sometimes when we do extraction, we might need to use some solvent and it consume a lot of time. So the reason why here artificial here is coming in is that we are trying to use the image. Okay, we are trying to use the image capturing using a camera and we use that image to determine what is the condition of the microalgae. Okay. So basically, with that, we are able to know what is actually going on with the cultivation and with the growth of the microalgae. Okay. So without further ado, I would like to introduce what is actually microalgae biomass for everyone to know. Okay. So what one need to know about microalgae is that they are known as third generation biomass. Okay. The second generation is what we call the lignocellulose biomass, and the first generation is called the edible food biomass. Okay, because of that controversy between first and second regarding food versus fuel, therefore we move towards a much more sustainable biomass, which is using microalgae. Okay, so why using microalgae? Because they contains high amounts of proteins, lipids, and carbohydrates. And all these kind of composition, right, the feedstock, they can be converted into other high valuable compounds, such as biofuels, supplements, animal feeds, biofertilizers, biochars, and many more. Okay. And what makes microalgae so special is that they can undergo a process which is called photosynthesis process. Okay. So basically, photosynthesis process is what plants organism has, where they can absorb the CO2 from the atmosphere, right? Atmosphere. Therefore, with the presence of UV, perhaps we can get this from the sun. What we have at the end of the day is what we call the sugar plus oxygen. And these are what we call source of energy, a very basic photosynthesis equation of it. Okay, because microalgae can consume carbon dioxide, and this is one of the things that we are trying to tackle in the current research initiative, because we want to lower down the greenhouse gases in the atmosphere, and it's also one of the initiative of low carbon. Okay, so that what makes microalgae a much more renewable biomass of feedstock. Okay, so for those participants that have not seen what does the microalgae looks like, you can take a look at the picture here. Basically, it looks something like this. Okay, I believe you guys may have seen it before previously. So it looks something like this. Okay, this is the Haemococcus fluoris microalgae. It's a spherical in shape. And what makes them special is because they can turn from green to red color. And the red color is what we call the astaxanthin pigment. Okay, so the second one is Spirulina platycis microalgae. And this microalgae, it contains a carotene, which is called C phycocyanin, okay? Or in short, we call this CPC, okay? And this is a blue pigment, right? It's a blue pigment protein, all right? And we have also the chlamydomus, right? The chlamydomus, this microalgae contains a lot of lipids. They generate a lot of lipids, and we know that the lipid can be transesterified into biofuels. And last but not least, we have is chlorella. All right. So chlorella is very strong microalgae because they are used, right? They are used for waste, water, bioremediation. Okay. And what you need to know about this kind of microalgae is that they can consume nutrients. Okay. They need the nutrients to grow. For example, the plants, the terrestrial plants, they need nutrients. What kind of nutrients? Carbon, they need nitrogen, and they need phosphorus. okay? The basic kind of nutrients. Of course, they will need some micronutrients as well. All right. So this is somehow a very brief introduction that we have covered in our first workshop on the upstream and downstream of microalgae. And for those participants who are new here today, I'm just going to very brief through how does microalgae works and what are them and what do they look like. Okay. So moving on, today's topic is on the artificial intelligence on how we're going to use AI for microalgae biotechnology. Because we know that the microalgae, they have different kind of species, okay? And not just different kind of species, is that we need to identify what are the species out there. Of course, from here, the statement says that it consists of 40,000 to 50,000 microalgae species. All right. Just imagine if for us to isolate each of the microalgae species from the fresh water, and then we do the DNA extraction just to see and to see what species is it. So it consumes a lot of time. Okay. And that's the reason why we are trying to implement the AI into it. Okay. So as you can see, microalgae, they have different species. They have different kinds. We have different shape, right? We have different shapes, different size. Okay. We have different size and different color. Okay, so because of the varieties of the microalgae species, therefore, we need to find a very efficient way on how we can actually isolate them and how we can actually characterize them. Okay, so moving on, the conventional quantification method, right? So last Tuesday, we talked about the downstream processing. How are we going to analyze and quantify the microalgae biomass? Okay, so these are the conventional method, meaning that once we grow our microalgae biomass, the first thing we need to identify is the biomass concentration. Okay, and the most easy way to do this is to use the UV with spectrophotometer, right? And if you use the UV spectrophotometer, what we have is to get the value of absorbance, okay, at a specific wavelength, okay? So if this is the method, somehow it may be very complex, okay? And sometimes it may not be that accurate because if you have different model of the equipment, therefore the absorbance or perhaps the wavelength may not be same, okay? The accuracy of the equipment may differ accordingly, all right? At the same time, when we actually do other kind of quantification. For example, let me give you, let's say if I want to measure, okay, the concentration of carotenoids. Okay, let's use HPLC, right? High performance liquid chromatography. And if we put in our sample, first thing, what do we need to use? We need to have a mobile phase, okay? A mobile phase to transfer the solute to the sensors to detect, right? To detect the compound. The second one, what do we need? We need to have stationary phase, right? A stationary phase or perhaps a column, all right? And this column, right, is also very expensive to purchase. And the third one, what do we need? We need to have solvent, and this is HPLC grade. Okay, so basically, to measure the carotenoids, you can see that it involves a lot of material. We need to understand what is the mobile phase composition, whether the mobile phase, whether is it polar or whether is it non-polar, whether is it hydrophilic or whether is it hydrophobic. Okay, so basically we need to know the basic chemistry of not just our compound, but also we need to understand the equipment itself. How can it, detect the sample that we are putting in? Okay, and this eventually costs a lot of time to study about it. Okay, and the second thing is that not just that, we involve a lot of costs. We need to purchase the right column, whether our column is it suitable to determine our sample, and whether our column, right, how long that does it needs to be purged to clean out the system itself before we put in another sample. And same goes to the solvent use. It needs to be HPLC grade. When we talk about solvent using HPLC grade, it's very expensive. Okay, and not all of the campus or not all of the laboratory has HPLC, because to buy or to purchase the equipment from Agilents, right, is very expensive. Okay, so it's not that accessible to everyone. And that's the reason why we come to the topic here today, which is on using artificial intelligence. Because we know that AI right now is a very hot topic. Okay, so how are we going to use AI-driven method to actually analyze our data? I believe you guys have heard of this word, Python programming, right? Programming. Basically, when we talk about Python programming, is that we need to understand about some of the coding. What we need to input into the Python programming is what we call here data set or image processing, data processing or image processing. Data processing is more towards numerical, okay, numerical data set. Whereas image processing is more towards image using photo, all right, photo, right, as the data set. So what kind of machine learning model? We have some deep learning model, right? We have also some machine learning model like SVM, ANN, KNN, K nearest neighbor, okay? So this kind of approach is much more efficient, okay? And it's the non-destructive approach, okay? So what is actually non-destructive? We are not destroying our sample, right? But what we do is that we obtain one of the value through image, okay, or one of the data using the image, or perhaps the
Ibunya
numerical data
ibunya
set. So therefore, we put into our machine learning model, it gives us the data very quickly. So it's a very fast analysis. It provides a very good optimization because what we are doing is some basic hyperparameter optimization. And it's a very simple process. Okay. So the next one is on conventional microalgae identification. So in the earlier part, I mentioned about quantification. In this part, I mentioned about identification. So the keyword here, right? The keyword here is how are we going to determine the species of the microalgae? Okay. So this is slightly different compared to quantification. When we talk about identification, is to identify the species of microalgae. Okay, to identify the species microalgae, because we know there are more than 50,000 species of microalgae. Okay, so how are we going to identify each and every species of it? The conventional way of using it, the most what people are using, I mean, to identify the most common one is using DNA analysis. Okay, but the thing about DNA analysis is that it consumes a lot of time. All right, not just to isolate it, we need to do a lot of lab work just to extract the DNA of it. Okay, So it takes out a lot of time. And not just taking up a lot of time, it's very expensive just to run a single sample. And let's just say that sample is being contaminated, or perhaps the sample has been destroyed over time, right? So therefore, it may prone to some laboratorious errors. Okay. At the same time, to use this kind of identification method, the train, the technician also needs to be trained. He needs to be well acknowledged of the equipment itself. Okay. And I believe that not many people is an expert in a specific equipment. Okay. Of course, we know the basic of it, but to actually operate the equipment itself is very difficult. Okay. So moving on, we have also the light microscopy to see the cell very easily. We have also SEM to see the surface morphology, okay, see the surface morphology. Same goes to ETM, okay, and then we have also atomic force microscopy. So these are the types of conventional microalgae identification, right? But at the same time, why not we just use AI implementation, right? Using a digital microscope, right? A digital microscope, what we need is only this is the camera. So we take the camera to capture the photo through the digital microscope. And that is where we get our images. What kind of images you can see from here? The images that we can get something like this. And these are all using the digital microscopy. Okay. And then from there, we need to process our image. And then before we put into the image classification, and we use the machine learning model to identify the condition of our microalgae. So this gives a very high accuracy, but of course, when we talk about high accuracy, we need to have a large data set. Okay, it's not just that using AI is good, it's very reliable, but the data set needs to be a lot. Okay, if we have a small data set, therefore the accuracy might be much more, higher. It might be higher, but it's too perfect. Okay, we need more data set so that the machine learning model can identify which image or which data set, or to standardize the data set accordingly. Okay, so it's non-destructive. We can still preserve our sample. It's very good in optimization, and it's very fast and a very simple process. Okay. So I'm going to share with you guys what is actually using image processing, or what is image acquisition. What is stage one? All right. The stage two is image pre-processing. What is actually image segmentation? All right. And last but not least is image classifier. Okay. Basically, how are we going to use image as a data set so that we can use for microalgae quantification and also classification? All right. So moving into this slide, I would like to share with you what is actually image acquisition. The word acquisition is also what we call collection. Okay, how are we going to collect our data set? Collection of data set. Okay, so how are we going to collect our data set? All right. So first thing first, we need to have a digital microscope. Okay, you can have a light microscope. You can have a light microscope. But the second thing you need is a digital camera. Okay. So with that digital camera, therefore you will have your digital microscope equipped on top of it, mounted on it. So with here, what we can do is that we can capture image. We can capture image of the microalgae. Different size, different shape, different colors, all right? As long as we have the digital microscope, we can capture all this image. And basically, all this image is what we call, the keyword here, is called data set. Okay, clear, right? Data set is basically what we need, which is our input. Okay, which is our input, right? So basically, the definition of image acquisition is to capture visual information from the physical world, right? The physical world, and convert them into digital image that a computer can process. Okay, so these are all the digital image that we capture using the digital microscopy. Okay. The second one is that once we capture the image, we have a lot of image. We need to do image pre-processing. Okay, we need to filter the types of feature we need, right? So pre-processing is also known as filtering, okay? Or we call it in the exclusion and inclusion. Okay. So what type of image you want, right? What kinds of image are you collecting? Whether your image is it clean or is it contains a lot of noise, all right? Or whether the image that you capture inside there is a lot of dust, okay? So we don't want that. We definitely do not want this kind of dirty images. So that is the reason why we need to go for image pre-processing. Basically, the definition of image pre-processing is to enhance, right? We want to enhance the feature of the image, right? We need to make sure that the image is remove or suppress all the unwanted disturbance, right? So that our AI computer vision model can work effectively then to improve the data set. Okay. So what it does is that it transforms the raw image most of the time that contains the noise background, right? And some of the missing value or inconsistent value, right, into high quality image data set. We want the high quality image. If we don't have the high quality image, therefore it will affect our result. That means which is our output. Okay. So this is on image pre-processing. So what kind of image pre-processing that we have? The first one that I would share with you is on image resizing and image cropping. We capture a photo, right? For example, we capture a photo and the size of the photo perhaps is 3,000 times, and then from here is 6,000, 600, sorry, 300 and 600 times, okay? So basically, let's say if we have a single photo, we might need to crop. Inside the photo has some important component, all right? Some shape, all right? But what we actually want, right? What we actually want is only the star, right? So we can crop the image accordingly, right? Which mean that we only want this part, right? Which is the cropped image that contains the star in it. Okay. The other one, the other type here, let me highlight this. The other part here, okay, the other part here, the yellow one highlighted, this is actually what we call, perhaps I use a different color. This is what we call noise, or we call this uh, disturbance. Okay, these are not what we want. All right, we only want the star, and this is what we call good quality image data set. Okay, so we can do this through image resizing and also image cropping. All right, I believe most of the participants here, you will have the Instagram or the Facebook or maybe the TikTok. You will edit your video, right? You will edit your video. You will try to resize and rotate your image that you capture. So it's the same process that we are trying to do in this capturing the image, okay? Resizing and also cropping to the shape that we want, okay? So moving on, we have is image grayscaling. So what is actually image grayscaling, right? It's actually to define your image when you capture, you will do some editing, okay? Editing of what? I believe the female participants here, right? When you capture the photo, you will edit, right? You will edit the photo based on the contrast, based on the color balance, the brightness from your background. You want to have a beautiful, beautiful image, right? You want to make sure that your skin tone is very fair. You want to make sure that the color balance between your face and your neck is similar, right? So that is where you do editing, right? So basically, grayscaling is a process where you convert the image that you have taken into various color spaces. So what are the image scaling out there? For example, you have RGB, red, green, blue. Okay, red, green, blue, which is this RGB. Okay, and these are what we call the color machine learning, okay, depending on the color itself. So later on, we will look into this as well. And then you have the CMYK, cyan, magenta, yellow, and black. Okay, so these are also the types of grayscaling. Another one, last but not least, is what we call the HSL, hue, saturation, lightness, and brightness. Because the hue represents the spectrums of the rainbow colors. Okay, at the same time, the saturation depending on the intensity of the color you want. And last but not least is the light and brightness. How much light exposure you are giving to that image. Okay, so that is what we call image grayscaling. And moving on, we have another one which is called image denoising or image reduction. Once we capture a photo, definitely inside the photo, you will contain some of the impurities. And we want to remove that out because that impurities will affect our data set. Okay, so the keyword here, denoising, is to remove impurities. Okay, so the way of us to remove the impurities, right? This is conducted through the image capturing before it released into a single image itself. Okay, so for example, at this part, you can see that some of the noise image, and this is what we call piece, all right? Somehow it's some background noise that we need to remove so that we have a very high definition, right? A very high that high definition or the word HD, all right? HD image. I believe right now we don't use the HD image, right? Last time we used the 180p image, right? And I believe during our childhood we used the 30p, right? Which is very low image quality. Okay, but now they already have the 4K, the 4K TV, and also sometimes they have the 8K TV. So you can see that the resolution, all right, the resolution of the image increase, or we call the word enhance, okay? Enhance the image of it, okay? So this is where image denoising takes place, that it actually helps to remove the small, tiny impurities from the image itself, okay? And then we have image segmentation. All right, so here comes a very recent technology. What is actually image segmentation? The first one is that when we capture a photo, when we took a photo itself, the basic photo we have, okay, let me draw here. And this is the human inside the photo. Basically, this is what we call image classification, a very basic image, an image that contains the object, image that contains, right, the image, it contains object, it contains the background, okay? So the one that I've highlighted in yellow, this is what we call our object, okay? Our object, and behind here, all right? All the one behind here, the green color part, okay? These are what we call our background, okay? Our background. Basically, the image classification took a single photo and behind is the background. However, when we talk about segmentation, segmentation is a process to separate the background from the object. So right now, we want to remove the background, okay? So we only want the object. So how can we do this? There's a few methods you can see here. Localization, right? Object detection, and this is what we call image boundary box. Boundary box, okay? And this is B. The last one is what we call image instant segmentation or image segmentation. Okay, image segmentation is a bit special because you can see it draws the line of the object. It only identifies the object, whether is it a cat, whether is it a dog, or whether is it a duck. So it only draws, okay? It only draws line or capture the lines of object, okay, the objects only. So this is what it meant by image segmentation, okay. And last but not least, image pre-processing, we have one of the important feature, which is what we call feature extraction, right? The feature extraction and also the feature selection. So you can see that once you capture all your image data set, but we know that not every image data set it contains the information or data we want. Okay, so that is where we do some extraction. So you can see the definition of extraction is useful for situation where input data to an algorithm is too large, meaning that it contains a lot of redundant information. Okay, so let's say that the process of the extraction because our data set is too large. Okay, our data set is too large. It contains a lot of unwanted or unimportant data or information. So that is why we need to do extraction or filtering. All right. So at the end of the day, what we have is what we call selection. We only come up to the final output of it. Okay. So this is how we do feature selection. If it's red, the output will be red. If it's green, the output, okay, will be green. If it's blue, the output will be blue. And if it's navy, the output will be navy. Okay. So basically, we need to do extraction because if we don't do extraction, at the end of the day, you will see that you will have a different output. Okay. You have a different output, and that is not the color you want. So feature extraction, feature selection is that it helps you to identify which is the important data set to be placed in your input, okay, your input, and in the selection, it helps you to collect what is the correct output, okay, which from the data set of your input. All right, so this is called feature extraction and selection. And last but not least is on image classification. Okay, image classification. Image classification takes place where you put all your image that you have captured into deep learning model or machine learning model. And the machine learning model themselves, they will play their role to identify, right? To identify what is happening and how they identify or label your data set. Okay, so basically, this is how the step takes place from image acquisition, right? Image acquisition, where you take all your data to image processing. You need to process your data using image resizing, the noise, grille scaling, all right? And then you have image segmentation. And moving on with all your inputs, you will have image classification or image classifier using deep learning model. Okay, so this is how the step by step from taking your image, filtering your image, editing your image, all the way to the machine learning model. Okay, so we will look into what kind of machine learning model that I have carried out throughout my studies. Okay, so I will share with you the first one is on data processing. Let's say that if you don't have camera, but you have a lot of data. Okay, what kind of data set we are talking about? Numerical data set. Okay, for example, you have pH, temperature, you have what we call biomass concentration, you have stirring stirring rate, perhaps CO2 inlet, okay, or perhaps CO2 flow, okay. What else data you have? Light intensity, okay. What else you have? You have your um, chlorophyll A, chlorophyll B concentration. All right. What else you have? You have protein concentration. Number 9, you have carbohydrates. Right. Number 10, you have lipid. Basically, all these from 1 to 10, I believe you will have more than 100 data set. Am I right? And then from all these 100 data set, you will somehow have duplicate data. You will have triplicate data. From here, you will have your mean value. Okay. So basically, you can see from data processing using your numerical data set, from the growth of bioaccumulation, you will have a lot of data which can be used for the machine learning to predict the output of your condition. Okay. So this is one of the study that I've used the data set. Perhaps I will use another new slide just to show you how does it work. Okay. So let's say that you have a lot of data. Your data set are in the forms of numerical. All right. And I mentioned just now, we have a lot. For example, biomass concentration, right? We have pH, temperature, light intensity. You have your CO2, okay? So basically, what do we call this? We call this as, let me use blue color, inputs, okay? So basically, we put into our inputs, what do we want is that it will pass through a machine learning, for example, ANN, KNN, SVM, right? And C and N. Will pass through the machine learning, and then you will have one part which is what we call your output. For example, maybe your output can be biomass concentration. Okay, so basically, you need to have all this data that is ready before you have your final biomass concentration as the output. Okay, so moving on to here, this is one of the study where I use the cultivation of microalgae. And what I have with the data, I put into it. And that is where I get all the data set, all the results that's coming from. My output is only to determine the dry cell weight. Okay, DCW stands for dry cell weight. I only want to know if which which absorbance will allow me to have the highest density, or which day the cultivation will give me the highest biomass concentration at the end of the day. Okay, so basically this is how I use the machine learning for me to identify it. Okay, so this is how we do this. At the same time, we can use the growth condition, right? We can use the growth condition to actually identify the biochemical accumulation. For example, day one, maybe my biomass is around 0.1 gram, and 0.1 gram contains perhaps around 0.02 gram of proteins. So how about if we cultivate it all the way to day 15? So let me get around 2.1 gram. So perhaps my proteins will be around 0.1 gram of protein. Okay, so we can use this kind of method to identify the machine learning method. Okay, so with that, you can see that the data set that is produced by the machine learning model, it can give you an accuracy. And this is how we study the accuracy. You can see that if it's near to the regression line, therefore, you can see that your R square will be around, will be higher, okay? But one thing you'll notice here is that you see the data set here is so saturated. It's very saturated. So your R square will be low, okay? Your R square will be low. So the accuracy is not that good, all right? And of course, we need to put in our inputs. These are our inputs, okay? For example, HSL, and then we need to put in the absorbance and also the day. And this is how we can calculate our output. Our output is in the form of biomass concentration or the concentration of C phenolcyanine. Okay, so this is one of the way we can do it. And at the same time, because the machine learning model, all right, the machine learning model will have different parameter. Okay, don't worry about that. Later on, I will show you a video. How do we do the optimization? Okay, so the hyperparameters, for example, C, kernel, gamma, accretion, we can actually alter each of the range using the Python system. And from there, we can get an optimized condition. For example, 100 RBF is the best, auto for gamma is the best, and the accretion is 0.1. We can get a very high R square representing our data is reliable. Okay, so this is how we do optimization. Okay, so this is a video to share with everyone. You can see, right? You can read from here. This is the part, the load X input, meaning that load X, meaning that we are putting all the inputs into our Python programming. Okay. Don't worry about the coding because the coding you need is another whole new story. But if we have our inputs parameter, you can see here, X data, concentration, day, TP, TN, COD, right? Lipid, carbohydrate, and proteins. Our load Y inputs, this is Y inputs is actually our output, okay? Is DCW, our dry cell weight. So basically, we have all this data in our Excel. We put into our machine learning model. We only want to see what is the output, which is dry cell weight, okay? So we load the data, we load our data. You can see our data set. This is all our data set. Okay, you can see our data set is more around more than 100. I think it's around 300 here. Yeah, 289 here. Okay, so you can see once we generate the data, we run our data, it will automatically run. Okay, and this is the hyperparameter. You can see C, kernel, degree, gamma, and then we can start our image processing, our machine learning processing. Okay, so it starts to run. Okay, so it will start to study the data set that we put into it depending on the hyperparameter that we control in beginning. Okay, you can see these are all the hyperparameter that it runs that we program into it. Okay, and then after that, it will give us the output. You see, all these are all the output that has been generated. We do not even need to draw the figure, but the Python programming will actually tell us what is the actual and predicted data. Okay, so you see, this is the residue plot. This is how saturated from the data set. Okay, so basically, you can see all these kind of plots will be given using the Python machine learning model. Okay, right? So this is where we can see our training R square 0.9891 and our testing 0.98. So this is how we study for the data set. Okay. So moving on, we have another one which is image processing. Okay. So image processing is what we have mentioned. If we don't have the numerical data set, right, we can use the image to actually study the growth and also the bioaccumulation of our data. Okay. So I will put into 5 case study. Case study number 1 is to study the biomass concentration using the data. Study number 2 is that with the biomass concentration, I want to identify the C-phycocyanin concentration as well. Okay. Case number 3, which is on microalgae classification. Okay, meaning that with the image itself, I want to identify the microalgae species. Okay, case number 4 is on the image segmentation. So I will show you later what is image segmentation and also the boundary box, which is more accurate. And case number 5, which is on microalgae synthetic image generation, meaning that our machine learning model will also generate synthetic data set. And whether if this synthetic data set is it reliable to be used as our input data set, will it affect our output or not? Okay. So first thing I would like to share with everyone is on color feature-based machine learning. Just now I have mentioned with you on the image pre-processing, what are the second step? The second step of pre-processing is they need to filter the colors and edit the light intensity and the color intensity as well. So these are the three base color feature-based machine learning model. CMYK, RGB, HSL. Okay. So from here, what we can do is that we capture image. We can capture all the image from day 1 all the way to day 16. Of course, the concentration will increase. You can see that the concentration here, the concentration of the biomass will becomes very, very green, right? Will becomes very, very green. Okay. So from here, we will start to extract the color based on RGB, red, green, blue, HSL, and CMYK. And from here, depending on the color, we will put into the machine learning model, SVM, ANN, and CNN. We want to see which color feature-based learning model, whether RGB, HSL, CMYK, which one is the best condition. Okay, so you can see that we do not need to do any extraction, right? We do not need to do any extraction. What we do is that we only capture the image from the microscope using the digital camera, and from there we can get all the data through this machine, through the image processing method. Okay. But of course, inside this kind of model, we need to do some hyperparameter tuning. You can see this is the best condition, 100 RBF auto 0.01. Okay, once we do the optimization, and that is where we can determine our R square. You can see that the R square of HSL is the highest compared to RGB and CMYK. It gives us a 0.99 accuracy. Okay, it means that the output from the HSL data is a much more accuracy compared to the other types of color-based learning. So that HSL is a very potential color-based feature machine learning model. Okay, because why? It consider a few parts, it consider the spectrum of colors, right? It consider the intensity or contrast. It consider the lightness and also the brightness. So therefore, HSL is a very good color feature-based machine learning model compared to CMYK and RGB. Okay. So you can see that it gives us a very high R square value. And you can see that the saturation of the data is much more compact or near to the linear regression. As compared to here, you can see that the data set, they are very saturated and not forced on the linear regression. Okay. Right. So same goes to this. This is using multi-layer perspectron. All right. Multi-layer per spectron, multi-layer per spectron model. So you can see this depending on the hidden layer. Hidden layer meaning that we are increasing the level. We increase the level of the machine learning model, making it more complicated. Okay, so this is what we call the hidden layer. Okay, we are trying to train them. We are trying to train them from the inputs, putting into different kinds of hidden layer, all right, to see whether our output is it accurate or not, whether our output is it accurate or not. At the same time, you can see that HSL still gives the better accuracy compare to RGB and also CMYK. Okay, so HSL is a very good color feature based learning machine learning model. Okay, so this is how you can see the conclusion. HSL have a larger detection range, better accuracy, and this is very good to be used for microalgae biomass analysis because it gives not just the color, but it gives the specific color spectrum, the color intensity, and also the light and the brightness. Okay. So at the same time, we also study about augmentation. What is augmentation? Augmentation is a process where we try to rotate, to turn the shape, to turn the image of the data set. We try to do that, all right? And we notice that with the augmentation, the machine learning model is much more better compared without the augmentation machine learning model, okay? So at the same time, the second scope we look into if we don't have the money to buy the digital camera, okay, if the price of the digital camera is much more higher than what we call smartphone, all right? Because I believe the youngster nowadays we are using the smartphone much more commonly compared to use a digital camera to take a photo. So we are trying to see whether the smartphone can give a better image or not. So you can see from here, we compare using the digital camera. This is the digital camera, and we compare using this is our smartphone. This is our smartphone, and then this is our digital camera. So we compare whether which is much more better to see the accuracy of it. Okay, so we take a photo using the same color. This is the same color feature, which is CMYK, HSL, and RGB, and we use back these three machine learning model. Okay, this is machine learning model, CNN, XGBoost, and SVM model. Okay, so you can see that the results basically the digital camera, right? Digital camera it achieve digital camera it achieve a higher overall accuracy, meaning that the accuracy is very high. But the problem is the smartphone also achieve a competitive results compare using to the digital camera. It means that in the future, we do not purposely need to buy a digital camera to equip into our microscope. It means that we can also use the camera from our smartphone as long as the image dataset is comparable with the digital camera. Okay. So the third case study, because the microalgae, they have different shape and size. Okay, you can see here, microalgae, this is the three type of microalgae, Chlorella, we have Chlamydomonas, we have Spirulina. Okay, let me write it here, Chlorella, Chlamydomonas, and we have spirulina. Because they have different size, they have different shapes, and the color, all right, the color intensity is also different. Okay, different shape, size, and color. So how are we going to determine the different kind of species if we only take the images, okay? So we need to use some feature extraction. So if we are considering feature extraction based on the color, okay? Like for example, what are the color? HSL, RGB, and then CMYK. If we base on the color to determine the species, you can see there is overlapping. Okay, because we know that all microalgae, these three microalgae, Corella, right? They are all green color. Let me put the word, change the font color. They are all green in color. So it's very difficult for us to identify which is spirulina, okay? Which is spirulina, which is Corella. You can see that they are all overlapping together and the accuracy is low, okay? But if we depends on shape, all right, not color, but on the shape, whether is it square, whether is it circle, whether is it a triangle, or whether is it a star shape, okay? That depending on the shape. So therefore, it gives a better distribution. For example, this portion, let me highlight this in yellow. This portion, right? This portion of data, right? It belongs to what we call Corella, okay? And then this blue portion, right? It belongs to Clammy, all right? Chlamydomus. And the last one, and the last one here, it belongs to spirulina, depending on the shape. Okay, if we place based on the shape, then the accuracy will be high. Okay, so this is one of the way we can classify or to separate the different type of microalgae based on the shape. Okay, not on the color, but based on the shape. So case number 4, we talk about taking a photo, which is on the image segmentation. In the earlier, when we talk about image pre-processing, we talk about image segmentation. So for example, when we took a photo, we know that this is the object, all right? The cat is the object. But the problem is that the one at the behind, you can see that the one behind here, this this part, all of this part is what we call noise, okay? Or what we call background. And this is not what we want because we know that it will affect our data set. So the second one, which is on object detection or boundary box, okay? So when we talk about boundary box, it captures inside the box, okay? It captured inside the box. However, right, you will see one thing. There is still some noise, okay? There is still some noise inside the images. Perhaps let me put in red color. You see here, there are still noise, the grass, the trees, the skies, okay? These are still some partial noise, but it reduces compared to image classification. You can see that here are the noise, okay? Right? These are object, all right? Our object, all right? And last but not least is image segmentation, drawing lines at object or only highlight. It only highlights the object, okay? Highlights the object. So you can see that this part, they only highlight the cats, the cat's features. Okay, it only highlights the dog features. Okay, so what it does is that minimize it minimize okay, it minimize the noise of our data set. Okay, so this is what we want, the object that we want. Okay, so it minimizes the data set, so our accuracy will increase. Okay, so we did some studies. For example, this is boundary box. Okay, if we take the boundary box of it, you can see that there is still noise, the noise from the background. All right. At the same time, but what if we take image augmentation, segmentation, okay, image segmentation, we only draw the line, all right, the lines of the microalgae species, okay, segmentation. So this is boundary box. So you can see that the image segmentation, right, offers superior performance with a MAP of 0.950, which is much more higher compared to the boundary box. You see, it's much more lower. So it means that image segmentation is a good classification method. Classification method compared to the boundary box because it reduces the noise, right? Or the interference of our data set, giving us a much more detailed data at the end of the day. Okay. All right. So I think we are coming to the last one, which is on generating synthetic data set to use the data set as our input. Okay. So for example, this is our real data. Somehow, somewhere, the machine learning, they are very smart. They will generate synthetic data. Okay. So now we are thinking, if we are using this synthetic data set as our input, meaning that we consider them, we consider these data as our input. Okay. So you can see we taken the original data set. This is all the original data set. Original. Okay. You can see the original data set, right? We are still playing the image augmentation based on boundary box. So you can see that this kind of models, FastGAN, CFastGAN, GPixels, these are all the machine learning model. Eventually, they will generate a different set of data. Okay, so they eventually generate a different set of data set. And you can see that somehow the image will change in the resolution. You can see that the resolution decreases. Okay, but our input data set increase. Okay, our input data set has been increased, meaning that we are putting more data into our machine learning to see that the accuracy of it, is it better or not? So you can see that 100% synthetic data set achieve high accuracy of 82.67. Still, the original data set gives a higher one, meaning that we cannot use all the synthetic data set because somehow the accuracy will be lower compared to the original one because original will still remains legit. Okay, but somehow what you can do to increase the data set is that we can mix a combination of synthetic, right, a synthetic and original. somehow your accuracy will be around very high, 99%. So we can do some optimization to see, to put a ratio, not just 100%, but we can combine original and also synthetic data set, combine them together, and therefore this will improve the accuracy of it. Okay. So this is how we do the image processing. You can see that these are all the data set we have, iPhone 13, right? You can see all the image that we have taken, right? It's not just one, two image or 10 image, but with one image we collect is around 300 to 500 image data set, okay? So with all the data set we have, you can see that we take a camera photo. This is the camera digital photo, right? You can see all the data set that we have, okay? And then we put into our machine learning model, right? Of course, we do some coding of it already. We want to test on different machine learning color set, HSL, RGB, or the CMYK. So these are all our microalgae image. Okay, we capture a lot of image, right? You can see different shape and different size of the microalgae image. Okay, and then we generate different kinds of data set of it. These are the different images of it. Okay. So yeah, I think that will be all that I will be sharing with you. Specifically today, I'm sharing on the Industrial 5.0. How can microalgae incorporate with the artificial intelligence, and how we can use the artificial intelligence to determine the quantification, to determine the biomass concentration, and also the classification of the microalgae species. Okay. So with that, I'm thank you everyone to the participants as well as my supporters, my mentors, my teams. They have provide me the support. All right. So with that, I would like to open the floor for any Q&A. If you are unsure of what is machine learning, please do not hesitate to discuss with me at this time. Okay. So thank you, everyone.
Okay, thank you so much, Mr. Cole. That was a very inspiring session. I'm now currently thinking about some ideas in my head inspired by your presentation. And a while ago, Mr. Cole already mentioned about the AI that is being a hot topic nowadays due to a conventional method that has a lot of issues. And this AI could be used as a quantification and identification. Uh, and there are some completed research that has been done by Mr. Ko, such as data processing using a numerical data using Python, and then image processing to quantify algae concentration, CPC concentration, classification, and also detection. And last one is augmentation using a camera phone and digital camera. So now we're going to the discussion session. I welcome everyone, all participants, to ask question to our expert here by raising your hand. Okay, is there anyone who want to ask question?
Yeah, I think it's a very advanced topic.
Yeah, yeah. Some of our participants here are new students, the first year student, so maybe a
Yes, yes.
little heavy for them. Okay.
Yes, yes. Definitely it's a little bit heavy because it's a very it's a very niche area using AI.
Yeah.
And right now, because this area is still developing, might
Mhm.
might raise a lot of question how we implement image as the data set.
Yeah. Okay, but we also have some last year students also here. Maybe anyone want to ask? Okay, while waiting, I also want to ask something, Mr. Ko.
Of course.
So, the first one is when you do a resize cropping, grayscaling, denoising, segmentation, etc., what application do you use? Is it simply Adobe Photoshop or is it a specific application?
Okay, it's a very good question. You can use Photoshop because they offers, as long as the image app that offers this kind of techniques, like reskilling or anything, you can use it. All right. For example, for us, we can also use Word, when we use the Word, then there is one cropping image. You can crop it to a particular size. You can, for example, the image you have is 300 time to 300. Okay, it's a square image. Okay. And then perhaps if you want just a small portion, you can just crop it according to the size you want because we only want the object. But we need to be consistent. If we are using one type of cropping shape, for example, 50-50, so we need to make sure all our data set is 50-50.
Oh,
Consistency is very important. Okay. But make sure that
okay.
the object is being captured. Like my object is microalgae cell, right? So I need to capture the microalgae cell. And I also need to make sure this shape that I've captured, the shape that I've captured, does it contains the whole body of the microalgae cell or not? Uh, for example, the hands, the legs, the head, it needs to be in there. We cannot crop just like maybe the head is missing or the hands of the missing. So somehow this will affect the machine learning to read. Okay.
Yeah. Okay, okay. Thank you very much. And after that, after the photo being processed, do you input it into a specific application, or do you yourself build your own
Okay.
application?
Basically, what we do is that our Python programming is already code. We already do our coding and already modify our coding based on these images. Okay, we already do it. It's like a try and error. And then what we do is that we create a folder and we make sure that our input are allocated back to our folder. Okay, and that what I can say the limitation of machine learning is that we do not know what is happening in the middle. Okay, let me share you one thing. So just now you see about this part. Okay, this part, for example. All right. We know what is our input, right? This is our input. Biomass, the image or anything. Okay, this is our inputs layer. But what we do know, what is happening is at this part. Let me highlight it in yellow. You know this part, you see all the lines here? You see all the lines here?
Yes. Yeah.
We will never know what is going on. And this is what we call the black box of AI. Black box. We do know what happened. We do know how they process our data. And this is one of the biggest limitation out there that people are trying to explore. What is actually going on? How do they actually process the data? And this is the word black box. Yeah, the black box. That's what we call this part. Yeah.
Okay.
Anything that is processed in the inside there, we don't know what is happening. It's like going into the black hole. It's very black. We don't know what's going on.
Yeah. Still a mystery.
Yeah.
Yay. Okay, thank you very much. Okay, we go back to the participant. Is there anyone who want to ask question to our speakers right now? You can raise your hand or maybe type your question in the chat box if you have any issue. Okay. Oh, so the participant is not only the students. We have some lecturers here too. We have Professor Didik here. Good morning, Pak Didik. And then we have Ibu Kustianti. And also, yeah, Rahim. Maybe Professor Didik wanna ask something. Oh, Haibah? Yes, please, Haibah, you can ask your question.
Uh, First of all, let me introduce myself. I am Haiba Fifa, and I am Baisun from Universitas Pendidikan Indonesia. My question is more about AI, since I am a layman from computer science. My background is biology, and I don't know much about technology. Um, from what we heard in a lot of social media is that there's a lot of discourse about AI, especially generative AI, being
Yes.
unsustainable, being cost a lot of electricity and a lot of water. Um, but then I've also heard that that is only generative AI. And since this is microalgae is for sustainability, my question is more of, is there different type of AI, and if the AI that is used here is sustainable environmentally, not just economically? Thank you.
Mm, Okay, to your question, perhaps I might not have the exact answer about it because I have not explored on the sustainability wise perspective. Okay, but what I know is that somehow somewhere um there is a lot of models out there already. Okay, like the one we use here, um, flash gun, G pixels, CNN, VQVAE. So this kind of machine learning model, eventually they generate this kind of synthetic data set or generative AI. Okay, so eventually they will somehow generate based on the user that create them. Okay, but we really have no idea because the coding that we use are based on open source. Okay, so in terms of sustainability, I would say it can helps microalgae to do the quantification much more easier with a single image, right? In terms of economical wise, I am not very sure because it involves a lot of data set. And if it involves of data set, perhaps we need to have a data set bank to collect all this data and to actually invest into a big data set bank. I think this is beyond our researcher motive. Perhaps some policymakers may have to look into that. Okay. So yeah, that's what I'm seeing. Mm.
Okay, Haiba.
Thank you so much for your answer.
Okay, we have another question in the chat box. What type of prediction model is most effective for estimating microalgae harvest yields and how accurate it is?
Okay. In terms of prediction model, that depends. Okay. We can have the machine learning model, and another one is deep learning model. Okay, which is the convolutional neural network CNN. Okay, that depends on how you do your hyperparameter optimization. Okay, and it also depending on the inputs that you enter into the Python programming. Okay, so I do not have an exact answer to tell you which model is the best, but still we need to do optimization. Like, but in my case, right, in my case, we use the multi-layer spectrum model that is much more better, or the CNN model is much better compared to KNN. Okay, so that depends, right, on your data set. Okay, hope this answer your question.
Okay, thank you very much, Mr. Khoo. Again, is there anyone who still want to ask some question? Yeah. Okay. Is there anyone who still want to ask? Okay, maybe uh, since some of the participant is a new students, a first year student, maybe they're still processing the information, but hopefully, yeah, it must be in it must be beneficial for them to learn this topic today.
Great.
Maybe we can just close this session. Do you have any closing statement, maybe, Mr. Ko?
Again, another closing statement. Huh?
Yeah.
Um, I would say that the AI is going to be there. AI is going to be there for a long time, whether you like it or not. And in terms of our researcher, I believe, or maybe not just researcher, but for educators like us, lecturers, we will encounter a lot of difficulties, especially when students using artificial intelligence AI to do their assignment. Okay. But what I would like to encourage the students is that yes, we can use AI, but we should not rely on them. We can use them as a tool, but we should not rely them as a live tool for them. Because at the end of the day, we are still human. They are robots. Okay, they are something that is non-living. So somehow to create something genuine, something original, it still comes from our human itself. Okay, so that will be my some of the sharing with the students. Okay, so hope you guys have a brief idea out there. Okay, so thank you everyone.
Okay, thank you so much, Mr. Ko. So, ladies and gentlemen, and all of the participants, all of the students, we have now reached the end of our second session in the Adjunct Professor Series with Dr. Kuan Ko. And today's seminar has given us valuable insights into how AI and predictive modeling can transform the field of microalgae biotechnology and making it more efficient, data-driven, and sustainable. On behalf of the Biology Study Program, we would like to express our sincere appreciation to Dr. Khoo for his inspiring and engaging presentation. We also thank all participants for your coming, for your attendance. And as a reminder, on the third and final session of the series will be held on November 14th, featuring Professor Topik Hidayat, who will share his talk on microalgae-based future trees, and along with Dr. Ko's discussion on writing review articles and academic collaboration with FPMIPA. And we hope to see you, all of you, again next week. So once again, thank you very much, Dr. Ko, for your
All right, thank you
presentation,
so
and to
much.
everyone. Yeah,
Thank
and
you
thank you very much
everyone.
for
You.
everyone. Okay, have a wonderful day ahead and stay inspired and keep learning. Good morning, everyone. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Porco.
Thank you so much for you.
Yeah.
Untuk yang belum mengisi presensi, boleh silakan diisi dulu presensinya. Ada berapa yang sudah mengisi presensi, Pak?
Baru dua-dua nih. Tadi
Ya,
banyak.
dikirim juga di grup aja mungkin.
Oke.
Tapi yang di grup tidak semuanya hadir sih, itu juga.
Tadi cuma 60, berapa? 65 tadi.
Hmm. Ya, kalau ya, thank you everyone. Saya Liv ya. Assalamualaikum.
Waalaikumsalam.